All right, let's try this again. We're going to do a screen sharing. I'm using one of these apps to record now. What is it? Panopto, Loom. They've all got different names, these weird startups. Anyway, I'm also recording a backup just on a normal mic recorder. So that was nice. I ended up recording last night, hour and 20-minute lecture. It went great. And then there was no sound. YouTube just shit the sound out. So we'll try this instead. Anyway, okay, so last class we were talking about flower parts, flower morphology, plant identification, how you need to know the parts of a flower. This recording came out fine. It's still up there on the YouTube. It's unlisted. You need a link to it. We go through all this stuff. We're talking about synapomorphies of angiosperms. We're focusing on angiosperms primarily because that's 80% of living plants. 80% of living plants you're going to encounter in the world are angiosperms. Let me just make sure that this is still... Is this still recording? Yeah, okay. I see a little wavy lines. You always got to make sure you see those wavy lines to indicate that you're getting good sound. So 80% of the plants you're going to encounter in the world are angiosperms. Okay? Any plant you see is either going to be one of 5 major lineages, one of 5 major taxonomic groups. Evolutionary groups as well. Monophyletic evolutionary groups. They're either going to be the bryophytes. Well, bryophytes isn't really even monophyletic. Don't worry about that. It's one of 5 major groups. The bryophytes, the non-vascular plants, okay, the mosses, liverworts, hornworts, okay? Everyone knows what a moss is. Liverworts, maybe not. They're also kind of weird. They're pretty cool. We talked about that of alternation of generations. Remember the bryophytes, especially they got that weird gametophyte stage, that haploid, only one copy of the chromosome, or one copy of the genome, only one set of chromosomes, that is one copy of the genome, haploid gametophyte stage. It's either going to be a bryophyte or it's going to be a lycophyte, like the spike mosses, which aren't a true moss. They have a vascular system. The lycophytes, Phylogenetic, that stuff, that stuff that looks like you see the desert lycophytes sometimes, and they look like someone threw out an old shag carpeting. It's like a decomposing rug. I've literally seen it. And I remember I was in the Baja Desert once, and I saw a Phylogenella species out there that was all dried. It looked like someone threw out a shag carpeting, but there's also another group of Phylogenella that grows in, tropical shady forests and jungles and stuff. Okay, so you got the bryophytes, that's one lineage. The spike mosses and lycophytes, that's another lineage. And you have the ferns, that's another lineage. That's a monophyletic group. And then you've got the gymnosperms, like the pines, the redwoods, the ephedra, aka Mormon tea, Welwitschia, ginkgos, cycads, that's gymnosperms. And then you've also got the angiosperms, right? So angiosperms comprise 80% of living plants, of extant plants. And so that's what we're going to be focusing on today. We're going to be focusing on classification of angiosperms, but also how life is classified. And this will affect the way you think about everything around you, okay? All the life around you, all right? And it's most of what we're talking about today is taken out of chapter 4 in this book, Evolution by Carl Zimmer. You should be able to download it. There's a Google Drive folder attached to the video description. And there was also one I put in the Drive folder for the class. Great book. It's in PDF form. I suggest getting a hard copy if you really want to go into it. But the PDF, of course, it's like a $80 book. So PDF is available if not. So this is what we covered last class. We're talking about flower parts, flower sex, what it means to be unisexual, how most flowers are bisexual. The vast majority of flowers are bisexual. They've got both the stamen and the pistil on it. Oftentimes they mature at different times to avoid self-pollinating, because self-pollinating, means low genetic diversity. It means they're going to be stacking up recessive alleles. Higher likelihood of stacking up recessive alleles, okay? Recessive, dominant, that whole thing, being diploid, etc. Okay? So this is all in plant systematics, all these diagrams of flowers, what it means to have ovary superior, ovary inferior. This is all important stuff to know, so that when you look at a plant, remember we grew plants by flowers. When you look at a plant, you know what to look for to identify it. these traits, knowing these traits and what all these different traits mean, will mean that you can go and look at a plant and say, oh, it's got a raceme, or oh, no, it's got a spike. The flowers are sessile on that inflorescence. And then you can, when you read a description of a plant in something like a flora, or, that might be having a spike versus a raceme. Two species in the same genus might have similar flowers, but one produces a raceme, one produces a spike. Okay, that like some of the agaves produce panicles over here on the right, and some produce spicate inflorescences, spike inflorescences. That's a big diagnostic factor for figuring out what species you're looking at if there's any confusion. Okay, you need to know the terms in order to discuss the differences. So that's what all this stuff is, right? Patterns of the mustard family went into this. Those traits that define what family a plant is in are called synapomorphies. That's a key word. You got to remember that. Synapomorphies, shared traits that are a result of shared ancestry, of being evolutionarily related. Okay? That's a key word. That's how we group plants into families. Anytime you meet a new plant, okay, well, first off, you should know which of those 5 major lineages it is. The bryophytes, the lycophytes, the ferns, the conifers / gymnosperms, or the angiosperms. Okay? Naked seed, gymnosperms, angiosperms, seed in a vessel, seed in a fruit. That's what an angiosperm is. Okay? You should figure out what any plant you look at in the world is going to be in one of those 5 major lineages, one of those 5 major groups. And once you do that, if it's an angiosperm, any plant you look at, you should try to figure out what family is it in, especially if it's a flowering plant. What family is it in? First thing you ask, what family is it in? Because that's going to help you deduce what genus it's in and what species. Okay, we also went through, let's see, where was it? Taxonomic levels, okay? Domain, kingdom, phylum, class, order, family, genus, species. What we're mostly concerned about, all right, for beginner botanists, for beginner plant people, is family, genus, and species. Maybe order a little bit. You'll start to see similarities among things that are in the same order, like members of the English ivy and ginseng family and members of the carrot family. They're both in the same order. Apiales, different families, same order. You'll start to see similarities in their flower structure and inflorescence structure, and that's what we look at. We don't look at leaves, we look at flowers, etc. Some plants you look at leaves. Leaves can certainly help diagnose what you're looking at, what species you're looking at, and there are certain plant families that have traits among the leaves, but overall you are looking at flowers and fruits. Okay, flowers and fruits are basically the same thing, the reproductive structure of a plant. Every flower, if it's pollinated, produces a fruit. Amazing how many people don't know that. Okay, so we talked about Linnaeus and his fucking tights and, the 1700s, and how he was grouping plants by flower structures, and how that turned out to be mostly right, because that's a evolutionarily conserved trait, whereas leaves can shift throughout a lineage, and a plant can change its leaves without there being many evolutionary repercussions. Say a plant is growing in a place, and that place, it's a relatively mesic place. It gets adequate amount of rainfall, and then the climate changes over thousands of years, and that place starts to dry out. A plant can change its leaves, produce narrower leaves, start producing hairs in the leaves in response to the drying climate, without there being many evolutionary repercussions. If it starts messing around too much with its flower morphology, okay, it's going to have trouble reproducing. So the flowers stay relatively similar, but the leaves can shift. That's why we look at flowers. So there's Physaria fendleri, Streptanthus. These are all members of the Brassica family. Again, this is all last class. Okay, so where are we today? We're looking at taxonomy. Okay, and we're looking at how we group things. Let's go through all this. Okay, taxonomy, living things. We already talked about this. We're focusing on family, genus, and species, maybe order a little bit. Every plant has a binomial scientific name. The name is always italicized. The binomial name consists of a genus name and a species name. Okay, like for humans, it's Homo sapiens. The genus is always capitalized. The species name is not. Okay, for example, Helianthus annuus for sunflowers. Helianthus is always capitalized. Annuus is not. Okay, that's a great way to really show someone that you don't know what you're talking about if you start capitalizing species names. I was doing it when I first learned this stuff, and someone, of course, took me aside politely and was like, "Hey, we don't capitalize the species name. It's not how you do it." And it's generally written in italics too. but really just focus on not capitalizing that species name. There are 70 species of sunflowers. So this is where it gets interesting. You start thinking about, okay, say you like a certain plant. Say you're interested in like just the common weedy native sunflower. Helianthus annuus, right? All Helianthus are native to the Americas, North America specifically, all right? Any that grow anywhere else and on either continent were brought there by people in the last couple hundred years. This is where they evolved. All right, so you'd say you're the one species, okay, but you're also curious what other forms that form that you know has taken in response to different climates, in response to different elevations, in response to the presence of a different herbivore, etc. This is where stuff gets interesting. This is where taxonomy and cladistics and the classification of living things and the way that we group them by how evolutionarily related they are, this is where it gets really interesting. Because you can look at a plant like, say, like Helianthus annuus. You look at a plant like Helianthus argophyllus down here, which grows in the Texas sand sheet. You can see that it's been through tens of thousands of years of evolutionary selection pressure to produce hairy leaves, those woolly leaves. That hair protects the leaves against drying out. It prevents the stomata from losing as much moisture, evapotranspiring as much moisture. It prevents the air, that hot, dry air on the South Texas sand sheet, from blowing across the leaf and pulling moisture out of those leaf stomates. Okay, this thing is incredibly woolly, but it's just a variation on a theme. It's a variation on the theme of a sunflower. So it's really cool to think about. And you can see how evolution has sculpted these different forms from a form that you already know. Okay, and you can do this with any plant. Say there's a particular tree you like, okay, and then you see another one that grows 800 miles away in a desert or in a swamp. You can see those different climates and landscapes have shaped different forms. Okay, different variations in the same form. Really cool stuff to think about for anybody who's biologically inclined, who's a biophile, who's just fascinated with life. All right, which most of us should be, because that's composes the living world that surrounds us. Okay. So there are 70 species of sunflowers, 70 different species of Helianthus. Each species varies slightly in morphology. That's the way that it looks and the form that it takes, having different leaf shapes, growth habits, life cycles. Annual, only lives one season. Perennial, it comes back. Herbaceous perennials, like the roots stay alive, the tops grow, die in the fall, die after the season's over, goes dormant for winter, but the root's still alive, then it sprouts again next year. Herbaceous perennial. Versus an annual. You get a lot of annuals in the deserts. There's a lot of annual sunflowers that just produce a ton of seeds, and then come up the seeds. The plant dies, and the seeds germinate next spring. After the birds get into those flower heads and eat some, and then like total slobs, spill a bunch of seeds all over the ground, etc. We already said this regarding plants. The taxonomic levels that we will be most concerned with will be order. Family, genus, and species. Below the level of family, there can also be subfamily, tribe, subtribe, etc. But we're not gonna worry about that. Okay, those are mostly reserved for very, very populated families, very species-rich families, families with a lot of plants in them. Okay, who have been very ecologically successful, like the sunflower family, 28,000 species, I think it's estimated at. Okay, all over the world, everywhere except Antarctica. Okay, if you've got that much species richness, that is, you have that many species in your family, okay, just going family and genus isn't going to cut it. You're going to need subfamily, you're going to need tribe. There's all these different taxonomic levels of classification between family and genus. All right, so anyway, plant families always end in ACE. That's another thing to remember. ACE. I used to pronounce it ACA. It doesn't matter. You can really pronounce, either one works. All right, my friends who speak Spanish, who are botanists, produce Asteraceae, the sunflower family, Asteraceae. All right, I know what they're talking about. It doesn't matter. Okay, anybody who's gonna correct your Latin is a nerd, and not in a good way. Okay, just ignore them. It doesn't matter. Pronunciation doesn't really matter. Okay, what matters is that the listener, whoever you're speaking to, understands and knows what you're talking about. That's what matters, and this is universal across. I mean, all languages, right? Every botanical Latin. Okay, if the Chinese botanists use botanical Latin, Iranian botanists use botanical Latin, Swiss botanists use botanical Latin. That's the beauty of the system. We use Latin because it's a dead language. It's not subject to modern nuance. Okay, you can pronounce it however you want. So anyway, ACA. So families end in ACA. Orders always end in ales, ALES, ales. Subfamilies end in oidea, OIDEA. So if you see something that ends, if you see a word that ends in oide, you know that's a subfamily. If it's a plant, if you know it's if it ends in ales, you know that's an order, etc. AC family. Okay, there are two major means of arriving at a classification of life. All right, phenetic and phylogenetic. Okay, phenetic just means you're classifying something based on overall similarities, not necessarily on how evolutionarily related it is. Remember, there's this thing called convergent evolution, where something like euphorbia and cacti, completely unrelated plants. If you go far enough back, we're all related, right? But for the most part, they're completely unrelated. Their lineages diverged millions and millions of years ago. Okay, totally different orders of plants. Cacti and euphorbia, which are, let's go back to them. Okay, are look similar from a distance, but they're completely unrelated. Okay, when you look at the flowers, you can tell they're different. But looking at right here, you got Myrtillocactus geometrizans on the left, Euphorbia ingens on the right. Both Euphorbia ingens I see referred to as a cactus all the time. Okay, it's not a cactus. It didn't evolve on the same continent as cacti. Cacti are only native to the Americas. If you see a cactus plant in Africa or in the Negev desert or in the Mediterranean region, it was brought there by humans in the last 400 years. Cacti are only native to the Americas except for one species of Rhipsalis, which was brought to the jungles of Africa by a bird probably 5 million years ago. And it's just that one species. It's an epiphyte. It's an epiphytic cactus. It doesn't look like this at all. Okay, so that's convergent evolution. Okay, since both these plants look similar, you can look at their flowers and tell the difference. But also, if you look at the spines, like the spines on a euphorbia are stipules. The spines on cacti are just leaves. All right, they've modified their leaves. When you look close, you can tell even if it's not flowering. But at first glance, to a lay person, they're going to assume it's the same thing. That's convergent evolution, and that's what phenetic classification is. Just grouping things together based on how they look. That's not what we want for taxonomy. Okay, when taxonomy first started, when Linnaeus was wearing his tights, and he was, botanists were sending him these herbarium sheets of pressed plants from all over the world, and he was looking at them, looking at their flowers, and grouping things. He knew, he just had the intuition to group these plants together based on how their flowers looked. And like I said before in last class, he ended up hitting the nail on the head. That was pretty correct. For the most part, since leaves can be so plastic, they can vary so much, flowers are what we look at. The reproductive structures are what we look at because they are the most evolutionarily conserved. Remember, you can change the leaves without there being many repercussions to the plant, but you can't really change the flowers too much. Okay, the reproductive structures have to stay the same for the most part. Okay, unless there's like a new genus evolving, it's a whole other thing. Whole speciation event. Just don't even worry about that. So phylogenetic classification is that which is based on evolutionary history or pattern of descent, which may or may not correspond to overall similarity, as we saw with cacti and euphorbia. Cacti and euphorbia look similar, but again, convergent evolution, two things evolving to look the same because they evolved in similar climates. Euphorbia evolved in the hot, arid climates of that euphorbia did in Africa, okay, where it was also getting munched on a lot. Remember, there's more herbivore pressure in deserts because there's not a lot on the menu. They're relatively barren compared to higher rainfall areas, okay? Cacti evolved in subtropical, seasonally dry forests and deserts of the Americas. And so because of those similar climates they evolved in, they both evolution selected for, or the environment rather, selected for plants that look the same. Armed defense, storage, succulent storage, and what else am I missing with cacti? Anyway, you get the point. All right. Oh, and dropping their leaves, right? Just succulent stems, dropping their leaves. Because leaves have those stomata in them that leak moisture. They take in CO2, they leak moisture. Okay, convergent evolution. Many cases of convergent evolution. There's lots of plants in the Chihuahua Desert that are moth pollinated, and they produce similar flowers that smell the same, even though these plants are not that closely related. All right, that's because moths have been selecting for what they like, for what they pollinate. So their moths are more attracted to flowers that produce a certain kind of scent that they can home in on, that smells very pleasant, very perfumey, kind of like jasmine, etc. Like a jasmine, like perfume alcohol scent. Okay, so convergent evolution, moth pollination is another example of convergent evolution. Okay, moth pollination being present in so many unrelated plants that grow in the Chihuahua Desert, like the evening primroses or the mandevilla, macrosiphon, or I saw a menadora, which is in the olive and ash tree family that produces moth pollinated flowers, etc. So phylogenetic is what we want. When taxonomy was first invented as a system in the 1700s, it wasn't based around phylogenetics or evolution. Evolution wasn't even a theory yet. All right, you would have been crucified for suggesting that everything wasn't just the creation of gahd. Okay, but they were grouping plants they already knew based on flowers. Okay. Then it turned out now, basically, all taxonomy is based around phylogenetic classification, how closely related things are, which is how it should be. That's a really cool way to group things, and it gets you thinking about all kinds of different things, variations on a theme, how evolution works, how plants speciate, how the climate shapes and sculpts different forms, different variations on a form. Okay, so today scientists group all living things according to how closely related they are. I just said that, i.e. plants that are in the same family. Okay, each taxonomic level represents a different level of evolutionary relatedness, okay? Order is a more broad circumscription, a more broad level of classification than the one below it, which is family. So order, family, genus, species. Remember that. Come up with your own acronym for it. Order, family, genus, species, okay? Only firemen get stoned, all right? I don't know, whatever works. Okay, since plants were originally just grouped together on how they look, today many plants have had to be reassigned to new genera. So remember that initially phenetic classification of things, grouping things close based on how they looked, that all changed. Well, first off, that was all the information we had to go on back, 70, 80 years ago, 200 years ago, whatever. But with the advent of DNA and polymerase chain reaction, okay, amplifying DNA so you can get a closer look at it, which was basically invented in the 80s. Caryophyllales was on acid. That whole story, driving down Highway 1, imagine the DNA spiral ladder becoming unzipped and rezipping. I don't know. He was a wild man. Kind of crazy. But regardless, I mean, I guess you'd have to drive down coastal California Highway 1 on acid. But he's credited with coming up with PCR, which is used in diagnostic testing today, forensic testing, testing for COVID, etc. So, with the advent of DNA, we were now able to compare genetic barcodes. All right, literally looking at the sequences of adenine, thymine, cytosine, and guanine. Okay, the 4 DNA base pairs, and seeing how those at a certain section of the genome compared to that same section of the genome on another species, and looking at the differences between them and seeing how much, what gene regions they shared in common, literally holding up one DNA barcode next to another DNA barcode and seeing how closely related they are. Okay, so that's what that changed it. That changed a bunch of, it turns out things that were grouped, like this happened with a lot of things that were in the lily family, like yuccas at one point were placed in the lily family. It turns out yuccas are not closely related to lilies at all. They're in the asparagus family along with agaves and irises and etc. Actually, I think irises are just in the asparagus order now. But anyways, asparagales and then asparagaceae. But either way, this had to be shifted around. Okay, usually flowers are a steadfast way to group plants, but with monocots, a lot of them, the flower traits for monocots are flowers generally in multiples of 3, parallel leaf venation, etc. So, and if you look at a yucca flower and some, other the flowers of some plant species that are in the asparagus family, you can kind of see, like, yeah, it kind of looks like a lily. It does kind of look like a lily flower. All right, so I guess I get it to an extent. There's other little subtle differences that you can see between those two lineages, the plants in the lily order and the plants in the asparagus order. Um, that really, you can look at them and you can still tell they're different lineages when you look up close. But for the most part, they do look a lot alike. DNA really solved that, though. You could see vast differences in the DNA, in the molecular synapomorphies. Remember, synapomorphies are shared traits. They are what you look for to figure out what genus or what family something is in. Okay? So this was a great example. So this is why, you'll hear botanists complaining, oh, they changed the name on that again. Well, they changed the name because it turns out this plant was thought to be in this genus, but it's actually not. Or this plant was thought to be in this family, but it's actually not. It's in this family, and the DNA proved that. We got it wrong because we didn't have DNA, 120 years ago when botanists through this plant, found this plant and described it as being a member of this family. Okay, a clade. This is another really important term to remember. Okay, a clade is an evolutionary group of related organisms. Okay, a clade can be a formal classification like a family or genus or order, or it can be informal, like some of the subunits of classification of very species-rich families, like Asteraceae, the sunflower family, or Fabaceae, the pea family. There's an informal clade, all right, a solid group of things that are all related to each other more so than they are to anything else that's outside of that group, okay, which is what monophyletic is. That's the meaning of monophyletic. There's a group of an informal clade called the Merbellioid peas in Australia. Really cool. They have pea flowers, but they're like day-glow colored. The leaves tend to be armed. A lot of them produce really toxic phytochemistry, okay, like the compound 1080, which is a poison that the Australian government uses to control invasive feral cats and foxes, which are not native to the Australian continent and have been causing literal extinctions of some of those cool marsupials. There's a compound synthesized by Gastrolobium, which is a Merbellioid pea. It's a genus of Merbellioid pea, and they put the compound 1080 in these little sausages and then literally airdrop them into the bush, into the scrublands and the forests of Australia. That compound is it's synthesized by, or the chemical, the analog is synthesized by members of the genus Gastrolobium, which is a Merbellioid pea. The tribes, I think it's Merbelliae and Bossiae, but there's not a solid formal designation for the Merbellioid peas. It's informal. It's tribe Bossiae and tribe Merbelliae. I don't know why no one's created a solid clade for those yet, but either way, that's an informal. It's a good example of an informal monophyletic clade. It's cool about that 1080 too. Is the marsupials, the native Australian marsupials, are not. They don't succumb to compound 1080, at least not very easily. I think many of them are resistant to it because that chemical compound, they've evolved with it over millions of years. And so they've evolved resistance. But mammals that aren't from Australia don't have any resistance to it. That's why it works on controlling cats and foxes. And with the cats, I don't think it really works that well. The feral cat problem in Australia is terrible. Anyway, so what does it mean to be monophyletic? All the organisms in that clade have a shared inheritance. Monophyletic is that's the desirable of the three different phyletic designations: monophyletic, polyphyletic, and paraphyletic. You only want things to be monophyletic. When we are grouping things into a clade, or whether it's a family or an order or whatever, they need to be monophyletic. We need to make sure that everything in that family is more closely related to everything else in that family than it is to anything else that's outside of that family designation, that box that we're drawing around that group. We want it to be a solid monophyletic evolutionary group. Okay, and that means all the organisms in that clade have a shared inheritance. So because all life on Earth is related and all shares the same common ancestor, all of life is technically a clade. If you zoom out enough, if you zoom far out enough on that taxonomic tree, that pyramid, okay, So we can apply the grouping of clade to any level of taxonomic classification, okay? Whether it's, class or division or order or family or down to genus, species, etc. Okay, so all that is required for something to be considered a clade is for the grouping, the imaginary box that you draw around a group of organisms, to be monophyletic. Okay, I cannot stress enough how important this word is to an understanding of the idea of evolution and inheritance. And it takes a minute to get at first. It takes a minute to understand, but once you grasp it, and these diagrams that I'm about to show you will help you grasp it. Once you grasp it, it'll shift the way you think about everything. It'll shift the way you think about your own species, the way that we evolved. It'll shift the way you think about how life evolves, about what it means for one thing to be related to another. It's a really important concept to thinking about the living fabric that you're a part of and that surrounds you. Monophyly, it directly applies to evolution and natural selection. Okay. Let's see another way to say it. Organisms that are in the same genus are more closely related to each other than organisms that are merely in the same family or order, and so on. Okay. So those are just talking about the different levels of classification. So you want an order to be monophyletic. You want the family to be monophyletic. You want, right, which as you go down, each is a smaller and more narrow unit of classification, of specificity. Okay. Genus is more specific than family. The synapomorphies that unite something in a genus are going to be more specified than those synapomorphies that unite something in the same family or order. In order to be classified correctly as a clade, a group of organisms needs to be monophyletic. Just said this, okay? So this is what monophyly looks like. You're drawing a box around all these things. Paraphyletic and polyphyletic, these exist because these are designations that, that existed before, for example, before the advent of DNA. Really showed us how closely related things are and why we had to move yuccas out of the lily family. Okay, this would be a good example of why we had to move. Both of these would be a good example of why we had to move yuccas out of the lily family. Okay, so right here that would be polyphyletic. Okay, so paraphyletic, and this is the way I remember it. Paraphyletic has an A in it. What paraphyletic means is that you're including a common ancestor, but not all of the descendants. Okay, and this is what you'll see in reference to, talking about reptiles, but not including birds, right? Because birds are technically reptiles. They evolved out of reptiles. So if you are talking about reptiles and crocodiles, reptiles to include lizards and crocodiles, but you're not including birds, you're not including all the modern day descendants. So paraphyletic, it's got an A. The meaning of paraphyletic, ancestor begins with A. The meaning of paraphyletic means you're including the common ancestor, but not all the descendants, okay? Polyphyletic just means you're way off. Like these things aren't related at all. You have two separate common ancestors, which you see here. So these are the taxa, the different organisms. Taxa is the plural of taxon, which taxon is a species. It can be a taxon, can be a family. A taxon can be an order. And same with this, DEGH. These could be species. They could be families, which would mean that C and F are orders, whatever. It just means a level of classification. If you don't get it, don't worry about it. That's not important. What is important is understanding what these diagrams mean and actually putting them into play. And I'll show you examples here. Okay, I'll show you an example of a polyphyletic designation in the cactus family that was revealed to be polyphyletic by DNA. These two species or two different clades groups were classified as the same thing. It turns out they're far apart on the phylogeny when the DNA breaks out a family tree. Okay. Not desirable groupings, polyphyletic groupings and paraphyletic groupings. Paraphyletic group, a group that includes the common ancestor, but not all descendants that derive from that ancestor. We just talked about that, okay? Like referring to reptiles and not including birds in that designation, all right? It'd be like including your siblings and parents, okay, for on a smaller level of specificity, talking about your family as an analogy, okay? It'd be like including your siblings and parents and grandpa, but not your cousins or their parents, okay? If you're talking about your own family and you're just talking about your parents and your siblings, that's great. If you're including your grandpa, but not including your dad's brother's kids, your cousins, or your dad's brother, that's an example of a paraphyletic grouping, okay? And this is the same thing. When Darwin came up with all this stuff, the tree of life, it was literally like a family tree can be applied to the way you think about a tree of life. Same thing, but on a much smaller unit, right? And much more recent in terms of time. That's the other thing, is you can generally think about these relations as kind of like in a span of time. We'll talk about that too. We'll get into that. Polyphyletic group, aka way off the mark. We just said that. That was this one. Way off the mark means, whoa, we thought these things were way more closely related than they are. They're not at all. It's not even like we're including the common ancestor, but not all the modern descendants. It's like these things are so far apart in the tree of life that to draw a box around all of them would be so broad that we're including things that don't look anything like the organisms that we're talking about, like yuccas and lilies. Okay, like they're so far apart. Like you'd have to for yuccas and lilies to be a monophyletic group, you'd have to zoom so far out and to go so far back at where they both diverge, those lineages both diverge from each other. You'd be including orchids in it, which don't look like lilies at all when you get to they're just so unrelated. That's kind of a bad example because some people think orchids do look like lilies. Anyway, the point is, hopefully you get what I'm talking about. You have to zoom so far out. Okay. So again, two separate common ancestors, just like we see right here, where C and F are the common ancestors and they're far apart. Okay. And if you zoomed far back out and drew the box around B, you could do that, but you're zooming so far out that now it's like you're including stuff that doesn't look anything like the taxa, the species you're concerned about, D or G or whatever. So if don't worry, this will sink in. You just got to be redundant with it. Okay. In contrast to monophyletic, polyphyletic and paraphyletic are both undesirable groupings. Why? Polyphyletic, incorrect grouping. Species are not as closely related as thought. This designation contains two separate common ancestors. Basically, a reiteration of what I just said. Paraphyletic, you've circled a monophyletic group over here on the right, J, K, and I. But since you've also circled taxon A, and you're including that in this whatever family or order, whatever you're referring to, but you haven't included B and all the common descendants of B, that's paraphyletic. Okay, remember, ancestor begins with A. You're including the common ancestor, and A is in paraphyletic, right? That's how I remember this. Paraphyletic versus polyphyletic. There's no A in polyphyletic. Ancestor begins with A. Paraphyletic means you're including the common ancestor, but not all the common descendants. That's how I remember it. Here's the example I was talking about. Archosaurus, crocodilia. These are all reptiles, but you're not including birds. Okay, and this is what the DNA reveals too. Birds are reptiles. All right, birds evolved out of reptiles. So the designation reptiles is paraphyletic because it includes crocodiles and lizards, snakes. That's Archosaurus, but not birds. You can see where the mammals branched off early on. It's like 250 million years ago, the synapsids, I think. Synapsids are those things that you'll see in museums sometimes. They walk on all fours, they're low to the ground, they got a big sail on their back. They look like dinosaurs, but they're not. They're actually ancestors of early mammals. So a cladogram, everything we've been looking at, these are all cladograms, all right? These upside down pyramids, okay? But there's numerous ways to draw a cladogram. All these things on the right here, these four different things are all the same thing, okay? They're all cladograms. They're all the same. They're all showing the exact same thing, whether it's a circle or an upside down pyramid or a pyramid or this little thing that looks like a bunch of tuning forks branching off of each other. A cladogram or phylogenetic tree can be thought of as analogous to a person's own family tree, like that thing I just said about, your grandpa, including your grandpa in a family. Look at a literal family tree of your grandpa and your parents and your siblings and your cousins, etc. Okay, it's literally just a microcosm, just a smaller example, a smaller sampling of a larger group. Okay. That's all a family tree is. You can take the family tree as a perfect analogy to what we're talking about here. All right. The four diagrams on the right are different ways to display the exact same information. How closely related whatever organism or group of organisms represented by the letters is to another letter on the tree cladogram. You can see D is more closely related to C than it is to A. All right, but see this node where C and D branch off from each other? All right, if you were to flip that around, D could be right next to B too. So it doesn't, and you can do that. You can flip it, if you were to flip it around 180. So D and C are both equally related to B. Okay, it just means they branched off. And that's what this shows too. It's like D and C both, this thing forked, one lineage went towards B, one lineage went towards C and D, this circular cladogram here. But either way, D and C are for sure more closely related to B than they are to A, because A branched off this pyramid thing and both the circular thing much earlier. And you'll see that in the traits, the physical traits they share and the DNA, the molecular traits they share. When you hold the DNA barcode of taxon C and D up next to taxon B and taxon A, you'll see that they have more base pair and code, the literal code, the A, T, C's, and G's for DNA base pairs. They have more of those in common. The code is more closely aligned with B than with A. Still not the same because they're different taxons, different taxa, they're different species. But all right, in each drawing can clearly be seen that species D is more closely related to species C than it is to species B or A. Okay, just what I said, same thing. Okay, well, I was saying it's more closely related to B than A, which is also true, but either way, you can see D and C are very closely related. So that would be a monophyletic clade. D and C would be a genus. And then, anyway, okay, in some ways, phylogenies and cladograms can even be taken to represent a literal timeline of how an organism likely evolved. And you can do this with something like how whales evolved from terrestrial mammals. You can see up here at the top of this diagram, okay, 55, 50, 45, those are millions of years, MYA. So 55 million years ago, okay, we know this from fossils. There are fossils we'll find in a body of rock. We'll date the isotopes in the rock, radiometrically date the isotopes of the rock and what their half-life is and how they decay, and get a solid date within the range of a relatively small window of time, and know that this rock is 55 million years old. And we'll see, and then we'll find other fossils, another fossil over here and another fossil over here, indicating how whales evolved from four-legged mammals. Like you can see here, like Pakicetus was a four-legged terrestrial mammal that existed during the Eocene, roughly 45 to 50 million years ago. And you can see, looking at the bones and the similarities of the bones, and then how you find another fossil, and now the bones you could tell it's related. The bones look a lot alike, except they're starting to change. This thing is starting to lose its hind legs. It's showing more adaptations for living in water. And then you find another fossil, same thing. Okay, this is closely related. We can tell it's related to this, but now this thing is starting to lose its hind legs altogether. And the front legs, the forelimbs, are becoming fins. You can tell that the way the bones are splayed out, that they're starting to evolve into fins. And so this is what we've done. And so this is really illuminating. You can literally see how whales, over a span of probably 30 to 40 million years, how they evolved. And all these pink things right here are synapomorphies. This is a new synapomorphy that evolves. This pink bar that crosses the evolutionary tree right there, perpendicular to the lines, those pink things are synapomorphies. Something evolves and everything on down inherits that, like in involucrum, freshwater semi-aquatic habitat. Or it evolves a fat pad in the jaw for hearing, and brackish for hearing. Okay, fat pad in the jaw for hearing is occurring in brackish water habitat. Also, you can tell that from looking at the rocks, at these sedimentary rocks that these things are found in. Saltwater habitat, they can tell that obviously from finding different mineral deposits in the rock that the fossils are entombed in. Nasal opening shifted back, okay, like whales. Their blowhole is a nose. You can see this. You can literally see this in the fossils. You see the nose start to shift further back, as time goes on. And the younger rocks, the younger fossils, the quote more recent fossils, still 40 million years old, whatever, but you can still see it. Now they're evolving blowholes. Okay, the nasal passage is shifting back slowly over time. Remember, individuals don't evolve, populations do. These are all baby steps occurring over millions of years, being selected for by the environment, things that can blow if their nasal passage is up top of their head, that's going to be easier for them to exchange air with the atmosphere. They've just got to come up to the surface rather than stick their whole head up above the surface. You can obviously see why that would be adaptive. They're going to do better. They're going to thrive more. They're going to be more fit for that specific environment. Not necessarily strong, but more fit for that environment. Okay. I don't like that only the strong survive, because that's not correct. All right. What does that mean? Strong is a vague term. It doesn't mean anything. It's fitness. What is fit in a desert is not going to be fit in a swamp. Okay. Those are completely different habitats. What's going to be fit at high elevations is not going to be fit at lower elevations. All right. The pink bars represent synapomorphies. So the words we have to remember so far: clade, synapomorphies, monophyletic. Okay. It's just a zoomed up cladogram of that whale, that evolutionary transition from terrestrial mammals to whales. Really cool shit to think about. Okay. The method that groups organisms that share derived characters is called cladistics or phylogenetic systematics. Remember, phylogenetic, cladistics, same thing. Okay? You can see chimp, mouse, pigeon, lizard. There's a thing of all. There's a cladogram of all these organisms that we know. Maybe people don't know hagfish, but you know that they're basically jawless fishes. They're a modern member of a lineage that branched off from the tree of life before fish evolved jaws. Okay, cladograms can also be represented by DNA codes and changes in the coding of various genes. Okay, like I just talked about, holding up one DNA barcode to another. They literally call it DNA barcoding. My friend Alan Rockefeller is a mycologist. He started doing this stuff. Never went to school. Learned it on his own. Actually, I think he had someone at Berkeley teach him PCR over a summer, and he was helping them out and learned this. But he learned polymerase chain reaction. He got the thermocycler. He got the primers. All the things you need to tell that amplification when to start with the code and when to stop with the code and what specific gene region you're looking at. He did all this. All right, and he literally teaches DNA barcoding of mushrooms. It's really cool. He did this, and he found out that things that we were calling a certain mushroom in North America was based on a name out of Europe. But then when you look at the DNA, and they look kind of alike, but there's a little bit of inconsistency in how they look too. And that kind of got him thinking maybe these things aren't the same species at all. They're certainly closely related, but maybe they're not the same species at all. He looked at the DNA, held up the DNA barcodes, realized that the thing in North America that we were calling by the name of the same mushroom in Europe, not same mushroom, but similar looking mushroom in Europe, was actually a different species. As you would think, because the Atlantic Ocean is 3,000 miles wide, and these things have been separated quite likely for a long time. Spores can travel far, but the spore on a mushroom growing on a forest floor is not going to travel that far. And that's called biogeography. Okay? That is the study of where things evolve, and you generally see that certain plant lineages are indeed restricted, with a very few exceptions, to certain continents or geographical areas. Okay, like there's a whole suite of plants called the Gondwanan relics, after the supercontinent Gondwana, which was comprised of Antarctica and South Africa and Australasia, Australia, basically, New Zealand, Tasmania, and southern South America. They were all together at one point. So you get the same plant families that don't occur anywhere else in the world and nowhere in the northern hemisphere, like the Proteaceae occurring on Chile, or in South America. There's a few in Brazil too, occurring in South America, occurring in South Africa, and occurring in Australia. And that situation is repeated like dozens of times with plant lineages. And they got there as a result of being that old. The lineage is literally that old, from a time when all those continents were together before plate tectonics pushed them apart at roughly the rate that your fingernails grow over 80 million, 50 million, however many years. Okay. Biogeography. And that's what Alan was doing when he was, teasing out that, this is not the same species as the one in Europe. They're closely related, but, the species of mushroom. Look at the DNA, hold up the DNA barcodes. They would look slightly different, the mushrooms would, and that kind of gave the hint. And then when you look at the DNA barcodes, it's like, oh yeah, this is a different species. We've just been calling it that because no one's analyzed the DNA before. And anyway, so he's described the whole couple new species that way. Really cool stuff. This is also a cladogram. This fancy poster I got. All right. This fancy poster, it's a scientifically accurate cladogram. It's literally drawn as a tree of life. And there's old Charles Darwin's quote. There is grandeur in this view of life with its several powers, having been originally breathed into a few forms or into one, and that whilst this planet has gone cycling on, according to the fixed law of gravity, from so simple a beginning, endless forms most beautiful and most wonderful have been and are being evolved. Endless forms most beautiful. When I was getting into plants, and I was really nerding out on redwood trees because I was living in California at the time. And I could just see how big they could grow and they could live 2,000 years. And then I would go to the Wikipedia page and look at the taxonomy chart, which was kind of a mini cladogram, and see redwoods are in this family. What else are redwoods related to? They're so cool. I just have to know what else evolved somewhere else. And then I found out there's a Chilean species of redwood that's in the same family, Fitzroya cupressoides, also known as the Alerce. Most of them were logged in the 70s and 80s, but there's a couple old growth ones left. They grow in these really cool temperate rainforests that get upwards of like 4 feet of rain a year, but it's chilly like the climate of the Pacific Northwest. And that just blew my mind. I was like, if you go far enough back, Alerces and California coast redwoods, have a common ancestor. All right. They're both separate branches of this pyramid shaped fork, and I just thought that was so cool. And then you start thinking about all the other things that existed that have gone extinct, all the other branches on that pyramid shaped fork that would have come off that we only know from fossils, that we haven't even found fossils of yet, that we don't know. It's the imagination runs wild, and you start thinking about this stuff and what those forests look like. It's really cool. And also, how did they survive each in their respective habitats? Like, how did they, redwoods grow really fast because they literally sometimes grow in floodplains and also on very unstable slopes where they can get buried really quick. So they've got to get really tall really quick. Right. Same thing with metasequoia, the dawn redwood, which is only native to a small section of China, almost went extinct, and now is planted all over the East Coast. It needs a little bit more rainfall. It's related. And those are related to our own Texan Montezuma cypress and bald cypress if you go further east by Houston. You think about all this stuff, just seeing one form you're familiar with and then seeing what else it's related to. It's wild. Like knowing that pronghorn antelope are more closely related to giraffes than they are to deer. You'd see them and you'd think this thing is just a different kind of deer. It's not. It's actually more closely related to giraffes, which today are only on Africa. Wild stuff to think about. How life moves around, how it's evolved, why it occurs where it does, etc. Okay, and then over here on this, that's Charles Darwin's drawing. This motherfucker literally drew a cladogram 180, 190 years ago. Maybe it was maybe 160. I don't know. What was it? 1880s, 1870s. All right. That's the way when it was all starting to click, and it started to click because he was traveling around and he was seeing all these different continents with different life forms on them and noting similarities and saying, oh, he started to really, he zoomed out far enough due to his ability to travel. And, his I don't know, how'd they pack his ass on that ship? How'd he get on there? How do you get a ticket on the HMS Beagle? Anyway, he was seeing all these different life forms and really thinking about him, being like, okay, there's obviously something going on here. This thing grows here and this thing grows here, or if it's an animal, lives here. But I can tell they're closely enough related. Like they share perfect similarities in bones and in flower structures, but they grow so far away. So there's something there. They shared a common answer and it started to click. And so that's when he started. I mean, this is out of his notebook, this drawing. Dude literally drew a cladogram. He started to piece it together. So, anyway, you can see down here at the base of the tree, bacteria, archaea being the extremophiles that grow in places, like the hot springs of Yellowstone, hydrothermal vents. All right, this wasn't even known to be a separate clade until the 70s or 80s when DNA revealed that archaea were not just bacteria. They're extremophiles. They've got a totally unique genetic code. Though they share a lot in common with bacteria, they're actually a whole different evolutionary clade. And bacteria, obviously the things that make you sick, all right? When you eat some bad sushi, it's coming out both ends the next morning, all right? But our archaea generally are relegated to more extreme places like hot springs of Yellowstone, hydrothermal vents, etc. Then you got your eukarya up here, cells with the nucleus, more, quote, advanced life. You could see they're evolving later, 3,800 million years ago. That's 3.8 billion years ago. Okay, so there is a correlation of time. These evolutionary trees do imply time, but you got to remember what you're seeing alive today are modern members of an old lineage that branched earlier. They're not necessarily an old species or an old group. They're something that branched earlier. This is a cladogram too. This circle. This is a really cool cladogram from a paper that just came out in 2024. This is a circular cladogram that represents the phylogeny, evolution, and classification of all flowering plants. The dark gray ring in center, all right, the donut hole represents the Jurassic. And then as you move further away from the center, it gets younger. You go to the younger Cretaceous and so on. So that the age of the stems, where a lineage originates, which you can see if you zoom in on this, okay, the stems, so that the age of the stems correspond with the time period, okay? So where that node is indicates where it evolved, right? With the center of the donuts, the Jurassic, then you move through early Cretaceous, late Cretaceous, into the Cenozoic, which is comprised of the Paleogene, Eocene, Oligocene, Miocene, and Neogene. I don't even use that. I don't use Neogene. Holocene, where we are now. And so you can see, looking at this cladogram, this is really specific. The estimated origin, when this speciation event happened, and you get a whole new family or a whole new order evolving, a whole new evolutionary group. Wild stuff to think about, really cool. And anyway, so going back to that on angiosperms, something I forgot to talk about regarding flowering plants is that we talked about the 5 lineages of plants that there are, right? The bryophytes, which is actually paraphyletic, but it's mostly the non-vascular plants. Don't worry about that. Bryophytes, remember, mosses, liverworts, hornworts. What the shit's a hornwort? Most people don't know. You're probably not going to encounter them, but they're cool. Don't get me wrong. But anyway, I just know that they don't have plumbing. They don't have xylem and phloem. They're not vascular. You got the bryophytes. You got the lycophytes, spike mosses, lycopods, quillworts, stupid common name. Okay, so bryophytes, lycophytes, ferns, that's an easy one, right? And then gymnosperms and angiosperms. And each one of those major 5 classifications then has its own, number of lower classifications that they can be brought down into. All right, basically being at the top of the pyramid looking down, all right? You get into the top of the cladogram, the pyramids of cladogram, looking down into, there's within the gymnosperms, you have the pines and the needles and the ginkgo, which is its own weird lineage. It's alone now. The ginkgos are weird, motile sperm, swimming sperm, just like the cycads. And then you've got the true conifers, which is like the araucarias, the podocarps, the redwood family, etc. Okay, and I think pines are actually outside of that. I'll have to look at the old cladogram and the previous presentation, but I think pines are more closely related to ephedras and welwitschias than they are to redwoods. I don't know. Anyway, okay, but within angiosperms, the 2 major classifications are monocots and dicots. Okay, monocotyledon, a cotyledon is a seed leaf, 1 seed leaf, or dicotyledon, 2 seed leaves. Okay, really easy. Like when you grow a corn kernel and it germinates, you get 1 seed leaf. When you grow an agave, you get 1 seed leaf. When you grow a banana from seed, some bananas actually have seeds. The bananas you get in the grocery store are all the same clone, the Cavendish bananas, the same phenotype, okay, the same clone. All right, they're wild bananas. The progenitors of cultivated bananas actually produce seeds. All right, they're actually somewhat closely related to ginger. You can see that in the leaves. Okay, all the seedlings, monocots, have 1 seed leaf versus 2 seed leaves. Like if you grow a cactus seed, all right, it starts, within a few days, it just looks like a stem. But like at first, you can actually see 2 little prickles. It's cacti have 2 seed leaves too. Okay, monocots, dicots. All right, some examples of so that when you know something you're looking at, it's a flowering plant. All right, an angiosperm, which is more a newer lineage than the gymnosperms and the mosses and all that. when you know something's an angiosperm, this is the first thing you want to break it down to. Okay, is monocot or dicot. Really easy classification, right? Aside from having one single seed leaf, another trait of monocots is they've got narrow, long leaves, parallel veins. There's exceptions to all these, of course, but for the most part, this is pretty steadfast. Flower parts in multiples of 3, okay? Whereas flower parts of dicots generally in multiples of 5 or 4. All right, there are exceptions like the buckwheats, Eriogonum, right, is a dicot, but it's got 9 stamens. It's really weird. I don't know what that's about, but it's got 9 stamens, a multiple of 3, but it's a dicot. But for the most part, multiples of 3, irises, orchids, lilies, agave flowers. The flowers are on monocots have multiples of 3, 3, 6, 9, whatever. Generally 3 or 6. And when you do a cross section of the stem, the vascular bundles, the plumbing looks way different than the plumbing on a dicot. Okay, dicots and monocots, it's way different. Okay, most monocots don't have lateral growth. Okay, like a palm tree never gets wider; it just grows straight up. It doesn't have that lateral meristem. It doesn't have secondary growth. Okay, because it's a monocot. All right, no monocot has true secondary growth. Some, a very few exceptions have something comparable to that, analogous to it. But like pseudo secondary growth, but don't even worry about that right now. Okay, it's the point is there's multiple ways that these things look different. These two lineages, but yeah, that's another thing, that there's no true secondary growth in monocots. Okay, parallel veins, flowers in multiples of 3, 1 seed leaf, etc. Okay, also in their roots, monocots tend to have fibrous roots, dicots tend to have tap roots. Right, think of like grass roots. Right, if you ever ripped up invasive buffalo grass from South Texas, etc., it's got very fibrous roots. Right, and then when you get into it, because everything's more complicated than initially you think, when you get into it, see, these are all the angiosperms right here. Okay, and the green is the monocots and the red is the dicots, and you've got these things in between, the basal angiosperms, the more early evolving angiosperms. You don't need to remember this, but just know that it's not as simple as just monocots and dicots. Okay, these things, like some of the Piperales, that's cava is in Piperales. If you ever had cava, Winterales, this is mostly in the southern hemisphere. Very smelly, pleasantly smelly leaves. Okay, Laurales, containing the avocado family, Lauraceae, the sassafras family, magnolias. Okay, they're a more primitive angiosperm lineage. Right, not magnolias are more primitive, but the lineage is more primitive, and it doesn't really fit into the box of monocots or dicots. It looks like a dicot, but technically it's its own thing. Okay, so just know that, right? Nymphales, water lilies, ambarella, really weird plant that only grows on New Caledonia, which I've seen, really fucking cool, weird plant. And then down here you've got the conifers. So just know that it's technically not as simple for monocots and dicots, but for all intents and purposes, for us it is. And for beginner botanists and people that just want to know a thing or two about plants, it is as simple as just monocots and dicots. Okay, so in order to see how closely related one plant may be to another plant, we can look up a phylogeny of that plant's order, family, or even genus. It is often as simple as going to Google and typing in the name of the plant's family and the words phylogeny. So if I want to see a phylogeny for the cactus family, because I'm curious, for instance, what is sister to, which is a word that means what is most closely related to. Again, if you imagine a pyramid cladogram, right, the plant species at either end of that fork, those are sister taxa. Okay, whether it's a family can be sister to another family, or a genus can be sister to another genus, or a species can be sister to another species, etc. Say I want to know what the sister genus of Lophophora, the peyote genus, is. Okay, I type in Cactaceae phylogeny, and I'll get, and then I go to like the image search on Google, and I'll get a phylogeny of cactus, of the cactus family. All right, specifically the Cactoideae subfamily, which peyote is in, and I'll see how it's related to this plant called Obregonia, which looks nothing like peyote at first. It's got tubercles. It looks more like a flattened pine cone or a pineapple. But when you look at the flowers, you start to see it like, oh, that looks like a peyote flower. It's got a taproot. It's got a taproot too. Really cool stuff, okay? How those two evolved from each other and they share a somewhat recent common ancestor is pretty wild to think about, being that they look so different. But then you can start to see it in these little similarities, and you can see that with a lot of similar things. Let me see. Is there any pausing this? No, I got to wrap this up. So, okay, so let's do it for the mangoes. Mangoes are in the family Anacardiaceae. It's the poison oak family. It's the sumac family. Anacardiaceae is in the order Sapindales, which is named after the maple family Sapindaceae. Sapindales is the maple order. Okay, what else is in Sapindales? The mahogany family, Meliaceae. Okay, which really invasive tree called the Chinaberry tree is in Meliaceae. In Texas, at least, it's very invasive. And a citrus family, Rutaceae. So oranges are in the same order as poison oak, poison ivy, maples, and mangoes. Wild stuff to think about, but you can start to see it when you look closer, like at the flowers or some of the vasculature, the internal plumbing. Okay, so now we know that mangoes are somewhat related to citrus, mahogany, and maple trees, though the divergence between all these groups likely happened dozens of millions of years ago. All right, and then you've got this out group over here that's just something that we use when we're doing phylogenetic studies. But now you have a tree. So this is a cladogram for Anacardiaceae, and for mangoes specifically, it shows what they're related to, the genus Anacardium. And then up here you got Rus chinensis, Chinese sumac, and Pistachia, which is the pistachio genus. And right, this is fun stuff to think about. All right, it boggles the mind. It's interesting if you're interested in a certain. And you can do this with anything. If you're interested in a certain animal or a certain kind of lizard or a certain kind of plant, you can type in the genus, the family that thing's in, and type in phylogeny, and you will get this. You will get a cladogram, all right? In this case, the little tuning fork cladogram. But remember, cladograms can also be a circle or a pyramid or whatever, showing you what it's closely related to. And so things that are right next to each other, you could see and share the same branch, the same node and the same branch and the same stem are closely related to each other, all right? And you see here, maples are related to buckeyes in the genus Aesculus, etc. Okay, so it's really fun stuff to think about in a lot of cases. There's a zoomed in photo of this. Okay, here's a phylogenetic tree for the order Caryophyllales, the order that cacti, spinach, beets, quinoa, and jojoba are in. Okay, jojoba is like the plant that produces oil that's used in cosmetics. It's really good for your skin, so you always see the rich ladies using it and whatever. And it's native to the desert of the Sonoran Desert, right? Produces a little nut. You squish that nut, and cool plant. I think it's the only species in its family. Anyway, it's in the same order as cacti, spinach, beets, and Venus flytraps. Venus flytraps, Droseraceae down here, and the Nepenthes, the pitcher plants from Borneo, are in Caryophyllales, the order. This is a huge order. A synapomorphy for the order Caryophyllales is the production of betalain pigments. Okay, which is the color you see in beets. You'll see it in bougainvillea, which is a kind of a horticultural atrocity. You'll see it at Home Depot a lot of the time. They love planting it because it evokes images of Tuscany, whatever that shit, even though it's not native to Tuscany, it's native to the Americas. Okay, you'll see it like vining up a trellis or whatever. That same pigment is shared by all the members of Caryophyllales, the order. Remember, orders end in ales, except for like one or two families, like Phylogenetic and the other. They lost it. They evolutionarily lost it. I don't know why. Normally, red pigments are produced by anthocyanin pigments, or red color is produced by anthocyanin pigments in plants. Caryophyllales doesn't produce anthocyanins. They produce betalains. Why? What was the adaptive benefit? I don't know, because we don't say what did this evolve for. Nothing evolves for something. There's an adaptive benefit in producing something, and that's why evolution selects for it. That's the correct way to talk about all this stuff. But either way, you can see cacti, their sister family to Portulacaceae. Okay, Portulaca is a genus. There's edible Portulaca. I forget the common name of it, but and you can see it in their flowers, and Portulaca is also succulent, etc. All right, Phytolacaceae, the pokeweed family, pokeberry, whatever it's called. Amaranthaceae, spinach, quinoa, that are in Amaranthaceae. That's all the same order as cactus and Venus flytraps, etc. But you can see Venus flytraps are in this other clade, the non-core Caryophyllales from this one, the core Caryophyllales. And then jojoba is right there in the middle. Okay, And again, DNA backs this up, flower morphology backs this up. Here's a great example of the cactus family, and this is a great example of a polyphyletic, a genus that was classified probably 80 years ago before DNA that was later found to be polyphyletic. Okay, Pinocereus, the really cool cactus. They produce these big tubers. They grow in the Sonoran Desert. There's some that grow in West Texas. You won't see them because they blend in. They grow up through other shrubs, kind of like a vine. It's called being scandent. And you won't see them until they bloom, a big white flower at night, pollinated by moths. Some are probably pollinated by bats. And they've got that big tuber in the ground. So if it during the extra crispy times of the desert, which these things are certainly evolved to, they've been selected for over hundreds of thousands of years by these desert environments they grow in. They can die back to that storage tuber. It looks like a big bulb in the ground. The leaf just turns crispy, but it's okay because the thing is still alive in that storage tuber. Okay, Pinocereus is really cool. Pinocereus gregii, I think, is the one we get in West Texas. Anyway, these two, when you look at this, the things circled in red here are both Pinocereus, the genus Pinocereus, and it was determined via DNA. Well, these things are not that closely related at all. They share two separate common ancestors. Okay, all the members of Pinocereus over here have a separate common ancestor than this one over here, and there's a bunch of stuff in between that's not related to them, like Pagacereus, the giant cacti from Mexico, Carnegie gigantea, the saguaros, etc. So this is not right. This is polyphyletic, okay? These things were grouped together and considered to be Pinocereus because without looking at DNA, they do look kind of similar, but again, it was just convergent evolution, all right? Both bloom at night. Both of these clades, these groups bloom at night. They produce a stem that's generally spineless and just kind of like pencil thin in some cases and grows up through another plant. Okay, so they look a lot alike, but it turns out they're not that closely related. This Pinocereus down here is more closely related to Acanthocereus, which we get down here in South Texas as well, which has 3 ribs. This Acanthocereus has spines. It's also moth pollinated. And it tends to grow up scandently, leaning on other plants, on other shrubs and stuff. When you look at that, you can kind of see. You can see it. Okay, yeah, I get that. I get how that's definitely related. It's more closely related. I can see it. The DNA reveals it. You can see it. Okay, but this is a great example of a polyphyletic designation. So what happened here? Pinocereus was later transferred to the genus Acanthocereus after DNA. Because remember, we don't want polyphyletic groupings. We do not want polyphyletic groupings in our taxonomy. We want everything to be monophyletic, right? So that was grouped into a whole other genus, okay? And this basically explains it, okay? If we're confused about what polyphyly, paraphyly, and monophyly mean, we can consult this diagram. Hold on, I got to take a leak. I'll be right back. I can't pause the thing. All right, I'm back. Okay, so, and same thing here with this designation here. If we're confused about what polyphyly, paraphyly, and monophyly mean, look at this example right here. Here's a good example, all right? You can see monophyletic designation. This is just in reference to the apes, okay? Right here you have New World monkeys, Old World monkeys, apes, and humans. Over here you got lemurs, lorises, tarsiers, All right, what is a loris and a tarsier? Okay, these big-eyed things, they're probably generally active at night. They do look a lot alike, but it turns out tarsiers are more closely, well, they're actually more closely related to New World monkeys than they are to lemurs. Okay, and they're kind of in between. That's not a good, if you're classifying tarsiers, lorises, and lemurs all together just because they look alike, and they do, all right, cute little bastards, weird looking things. It turns out that that's a paraphyletic designation, because tarsiers are outside. Lemurs and lorises are closely related uh, to each other, but tarsiers are in between. They're an outgroup. They're more closely related to the actual apes and old world monkeys, new world monkeys, etc., than they are to lemurs. Okay, so that's a paraphyletic designation. If you're going to include tarsiers in the group of lemurs and lorises, you would have to include the new world monkeys, the old world monkeys, the simiiformes as well, including apes and humans. Whereas polyphyly is including lorises and tarsiers together. See, here's a loris on the left, a tarsier on the right, and they do look a lot alike, but it's not including lemurs. Okay, so there's a box drawn around lorises and tarsiers, but not around lemurs. Okay, so that just means way off. Two separate common ancestors. Okay, two separate nodes that are not included in the designation. This is a node, all right, where it branches. Okay, this is a really helpful diagram. All right, so you got to imagine that. Okay, polyphyly way off. Paraphyly, ancestor begins with A. Paraphyly has A. Paraphyly includes common ancestor, not all the descendants. And then there's another example showing the same thing. Okay, drawing a box around, this would be a polyphyletic designation, including mammals and birds and the same thing, but nothing in between. These things are way far apart. We're not that closely related at all. And if you had to include everything in between them, it would be meaningless, because you'd have to include crocodiles. They look nothing like mammals or birds. They're so far off. All right, you'd have to zoom down to such a broad circumscription down here to amniota. Okay, you'd have to be going so far back in the evolutionary tree in evolutionary time that you'd be including everything, and it just would be pointless. It'd be a meaningless designation. This is just redundancy. This is just reiterating what I've said. All right, here's a great example of DNA barcoding, of holding up different comparisons. Over here on the left, we have taxon 1, 2, 3 through 8. 1, 2, 3, 4, 5, 6, 7, 8. We're representing 8 different species or 8 different families. It could be any of those, but let's pretend it's species. And over here, we have different DNA sequences, 82, 082 through 123, 123. You can see they're basically all the same, except for sequence 115 through 120. You could see the GTTCAA code is the same through taxon 1, 3, 4, 6, 7, and 8, but then 2 and 5 are outliers. So 2 and 5 form a group. They're more closely related to each other than they are to the other species listed here, than they are to either 1, 3, 4, 6, 7, or 8. Okay, that's just great. This is literally a great example of how DNA barcoding is done. And you can see it, again, a change in the code, those 4 DNA base pairs, A, T, C, and G. And everybody should know those, the 4 DNA base pairs, all right, the four nucleotides, A, T, C, and G. We've all got them. This is just, again, different ways to draw cladograms, phylogenetic trees. The black bars represent synapomorphies, just like we saw on that whale cladogram. Okay, reconstructing phylogenies. This is out of Carl Zimmer's evolution book, chapter 4, right, which again you can download on that Google Drive folder in PDF form. I suggest just getting the book. If you have the money, you'll use it, and if you're interested in this stuff, you'll use it for the next 10 years of your life. Okay, because we did not directly witness species branching apart over millions of years, we must construct phylogenetic trees as our hypotheses about the real evolutionary history. To generate these hypotheses, we must gather data. All right. This is talking about comparing synapomorphies, comparing character traits. Okay. And so going on, it talks about, knowing, let's see, what is it? Comparing seals and dogs. All right. Carnivora species, dogs, cats, and their close mammal relatives, such as bears, raccoons, walrus, seals. Look at their skulls. We can see similarities. And so what are we looking for? We're looking for synapomorphies. Okay, again, just a reiteration, it's synapomorphies. Then you can see over here, taken from Carl Zimmer's Evolution Making Sense of Life. That's a great fucking textbook. Right? So how are the carnivorous species related to each other? We see these synapomorphies, that these shared traits, and then they're morphological at first, or at least when life was first being classified. But of course, we know, looking closer, it's all DNA. So if you want to read more about this, it's chapter 4 in Carl Zimmer's textbook. Great stuff. Okay, and there's taxonomy again. Just remember, we're only concerned with mostly order, family, genus, and species. And then as you zoom out, each level of each taxon, family, order, class, etc., gets more broad and it encompasses more organisms. Okay, it's literally zooming out on the tree of life. All right, taxonomic levels confer a degree of relatedness, how recently one species may have shared a common ancestor with another. Okay, and that's just talking about zooming out. Okay, things that are in the same order are not necessarily as closely related as things that are in the same family or the same genus, etc. I think I'm just repeating myself at this point. All right, these synapomorphies can be chemical, morphological. Oh, I already said chemical. There's a repeat, or molecular. Okay, so like a third of the members in the mango family produce urushiol, which is the compound that causes a rash in like 80% of the human population if they touch poison oak. Okay, urushiol evolved as an antifungal and anti-herbivory compound to deter fungi and insects. It didn't evolve for us; it just happens to cause a rash in humans, and so a lot of the plants in that family produce it. Not all of them, but a lot of them do. And so if you see, this can be helpful because if I was in Mexico walking through a cactus forest on this sketchy slope, and I saw this plant. I could tell it was in Anacardiaceae, and I just by the leaf pattern, it had pinnate leaves. That can be a lot of things that pinnate leaves. Members of the pea family, members of the creosote bush family, Zygophalaceae, but a lot of members of Anacardiaceae do too. And when I saw the inflorescence, it was a panicle with tiny flowers on it. I saw, okay, this is an Anacardiaceae. And then I saw that blackening on the leaves, which is what urushiol does when it reacts with oxygen. Boom, I knew not to touch this plant. I had a hint. This is probably what's going on. I'm not going to touch it. And indeed, later on, I found out it was. That was a cool genus in Anacardiaceae that grows in the desert among giant cacti and little peyotes and other little cool cacti. And it produces urushiol. Okay, so here's a great example. We're finishing up soon. Taxonomy. Okay, this is a Texas native Solanum rostratum. It's called buffalo burr. You can see every level of taxonomy for this plant down to species. All right, it's in the family Solanaceae, the Nightshade family, the order Solanales. All right, and you can do all this on iNaturalist. That iNaturalist app, you can click on the taxon page for a species that you're interested in, and you'll be taken. Or you click on the taxon name, it'll take you to the taxon page. The taxon being Solanum rostratum, and you'll click on the taxonomy, and you can literally see all this. This is why iNat can be so helpful, for learning. It's wonderful, all right, whether it's the app or the web page, whatever. And so you'll see, okay, this Solanum rostratum. And because Solanaceae is such a big family, it's got all these intermediate levels of taxonomy, like subfamily and tribe, and even subgenus. Between family and species, because there's so much in it. It's such a diverse family. All right, so here are some synapomorphies of the Nightshade family. We talked about this last class. The page on the left is taken from Botany in a Day by Epple. Great book, kind of a beginner book, but it doesn't even mention the word synapomorphies, but that's what it is. Those are the patterns. It says patterns. Every page says patterns of it says the patterns method of plant identification. Those patterns are synapomorphies. Just a technical word for those patterns, and it goes into the flower structure, and you start to see it. This stuff starts to click. Okay, and then on the right is plant systematics, a literal list of all the synapomorphies of the Nightshade family, right? Whether it's Brugmansia or Datura or nicotine or peppers or potatoes or tomatoes, etc. Right? So easy to read, be good for beginners. This is if you get more in depth, it really goes into the science of it. Right? Ovary position, how many stamens, corolla, 5 fused petals fused together to form kind of a tube. All right, and there's a written description of all the traits of the Nightshade family. Placentation, how the seed attaches to the ovary wall Perianth. Remember, perianth is composed of petals and sepals. This was in the last class. The flowers are bisexual, actinomorphic. That's radially symmetrical. Rarely zygomorphic. Sometimes they are zygomorphic, generally for hummingbird pollination or something else. But all these terms too become helpful, the deeper you get into this stuff. You'll be reading about a plant, you'll be trying to figure out what it is in a flora. How do I tell one species from another? You'll see a word in there like sympetalis. What the hell does that mean? Look it up in the glossary. Okay, it means the petals are fused together. Boom, that's super helpful. It helps to know this stuff. And so that way you're not just looking at pictures, you're actually getting into the nitty gritty, looking at the subtle details, okay? Taxonomy, order Solanales. All right, there's 5 families in that order. 5 different variations on a theme. Note the morning glory family, Convolvulaceae, is in the order Solanales too. And then you got family Solanaceae. Go down to all the different subfamilies of Solanaceae, okay? Cestroideae, the cestrums, the generally moth-pollinated, night-blooming, very pleasant-smelling genus of plant, cestrum. Okay, Nicotianoideae, all right, the nicotines, obviously. Petunias, Petunioideae, Schizanthoideae. Schizanthus is a genus of plants that produce really cool flowers in the Andes. All right, super diverse in the Andes. I saw a bunch in the Andes of Chile when I was there. Solanoideae, that's the genus Solanum. Potatoes, eggplant, peppers are in that, I believe. Is capsicum in Solanoideae? Probably, yeah. Tomatillos, tomatoes, etc. Okay, examples of synapomorphies of various orders and families. Every family and every order is going to have its own list of synapomorphies. Then you can literally type in, if you're interested, okay, how do I know something's a mustard? Type in Synapomorphies, Brassicaceae, the mustard family. Here's a list of some of them. 4 petals, 6 stamens, I forgot to put that there, superior ovary, and glucosinolates. That's that horseradish. It's like a sulfur. All right, kale farts. It's glucosinolates. That chemistry. All right, I think they're mostly anti-herbivory compounds. They probably confer some other kind of adaptive benefit, but it's a chemical synapomorphy for the family Brassicaceae. Onograceae, evening primrose family, 4 petals, 4 sepals, inferior ovary. That just means where the ovary is positioned. Is it below the sepals or is it enclosed in the sepals? Tomatoes tend to produce a superior ovary. Is that right? I didn't write that down here. But, you see the sepals on top of that tomato. That means that when the flower was open, okay, the fruit is above, the ovary was above the point of attachment of the sepals and petals. Superior ovary, inferior ovary, etc. That's in Plant Systematics. It's also in this PDF, which you'll be able to download. Ericaceae, the blueberry family. Lamiaceae, the mint family. This is immensely helpful because, again, when you're out in the field, you see a new plant, immediately the first question you ask is not what plant is this, what family is it in? Because that's going to be the first clue. Like you're trying to put clues together to figure out what a new species that you've never seen before is in. Synapomorphies of the mustard family. I literally just went to Google. I typed in Synapomorphies of the mustard family, and AI gave me this. All right, it mentions all the stuff we just talked about. And there's our native West Texas mustard. What was it? Hesperanthus. Anyway, linearifolia. I forget. I've got too many names in there. Follow-up questions. What are the 3 lower levels of taxonomic classification that we're most concerned with for the purposes of this class and for beginning botanists? Okay, family, genus, species. Which of the 3 following terms is the correct way to classify life and why? Monophyletic. Why did the advent of DNA analysis lead to certain plants needing to have to be placed in separate genera? Because plants 100 years ago, when they were being described, some of them 150 years, 200 years ago, depending on where you are, a lot of plants in the Americas were described 150 years ago, were originally grouped together on how they looked, not on DNA. DNA turned out that convergent evolution, or just sheer coincidence, produced plants that had similar flowers. Again, you look close, you look super close, you could tell the difference, but it was hard back then. So the advent of DNA really cleared up any confusion about how closely related one plant was or what family it belonged in. What is a clade? It's a monophyletic evolutionary group. What is the term that describes traits that are shared by all related members of a clade? Monophyly or monophyletic. Okay. Which of these 4 cladograms is the correct way to display a phylogenetic tree? Also, what's wrong with this question? Okay. They're all the same. They're all the same way to display a phylogenetic tree. All right. And what's wrong with this question is that it's they're all the same. All right. It implies that one of those is wrong. They're all the same. That's all I got. Hopefully you got something out of that. You will be quizzed on this. And yeah, check it out. Again, the books that I was using, Evolution, Making Sense of Life, Chapter 4, PDF is available in the Google Drive folder. Plant Systematics, for some of the flower descriptions. And then, of course, if you need a refresher, watch that first part of this PDF, that first class, okay, up here, where we're going through all the different plant families, okay, like, the carrot family. We talk about Azarella compacta, super cool plant. Some would not assume to be in the carrot family, all right, members of the pea family, the 3 different subfamilies. All right, Azarella compacta, the cool thing about that is, yeah, it can live for 2,000 years. It looks like this Dr. Seussian mat of green. Now I can't find it. Where the shit did it go? But this really gets into it, okay? This flower structure is how we grew plants. That's there you go. There's Azarella compacta. And great example of convergent evolution in this photo. That's Azarella compacta. This is a plant from the carnation family, Caryophyllales, also taking on the matted habit because it's growing at 15,000 feet elevation, where the winds can be brutal, where every night gets really cold. It's also at a low latitude. It's at 18 degrees latitude, so it can also be really hot. And they're exposed to more UV light, so they've got this thick, waxy covering. Okay, but they're from different families. Azarella's carrot family, this other thing, which I forget the name of, is in Caryophyllales, the carnation family. And then this is a little member of the Asteraceae, the Baccharis family. But yeah, this is all volcanic, 15,000 foot elevation landscape. All right. Hopefully this uploads and the sound didn't get messed up. Take care, and definitely email me if you have any questions about any of this. And please download those books and look into it. Take some notes, and this PDF will be available as well.